This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 159, recorded November 25th, 2011. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How's the turkey, Alan?
It was delicious.
Not sleepy, huh?
No, that's apparently a myth anyway.
Yes, I've heard. Tryptophan, is that what it's supposed to be?
Allegedly, yeah.
A legend.
Yes.
Also joining us today from North Central Florida, Rich Condit.
Howdy, gang. Except I'm not in North Central Florida.
Yeah, I can't really introduce you from Texas, though, right?
No, that's true.
Because you are indeed from there. But today you're in Texas.
Today I'm in Texas.
Visiting offspring, right?
Visiting offspring.
First and second generation.
Yeah.
How is everybody?
Everybody's great.
Yeah?
Everybody's really great.
Would you go down in the basement to do a TWiV?
No, I'm up in a. This is Texas. There are no basements. At least I haven't seen any. I'm up on the second floor of my daughter's mansion.
Why aren't there basements? Because the Texans likes to build up.
I guess I mean, there are no basements in Florida either.
Well, because they flood.
That's true. Everything's built on a pad. I don't know. You're going to ask the builders. Well, there may be basements somewhere, but there aren't any around here.
Well, I guess in Texas, you don't really need to dig down. You could just sprawl out.
Yeah, that's true. Or build up.
Yeah.
I don't know what basements are for anyway, except that, stuff doesn't freeze in the basement when it gets really, really cold.
In my experience, basements are there to leak.
That's right. They are.
That's their purpose. They're full employment for contractors.
I know. We had a basement in Buffalo. We had that same issue. That's right.
Well, I'm in a basement right now. I happen to be in New Jersey at home, so I didn't go in today. And my office,
we had one of those two-person swings, back-to-back swing on a swing that we used. We installed the swings in the basement from the rafters in the winter in Buffalo. That was great.
In the basement, you mean?
Yeah.
Yeah.
Things you do for entertainment when you get 6 feet of snow.
So you're in the basement in New Jersey?
Yeah, my office happens to be in the basement. So, I mean, it's very nice. It's all finished, but the windows are, up by the ceiling, and you you see the ground, and you look up. I always feel like I'm in a basement. That's it. It's okay, but I kind of like to be upstairs where there's windows and light.
Yeah.
But it's fine. It'll do. All right. Today we must be dedicated to do this, right, on the day after Thanksgiving.
Black Friday TWiV.
A lot of other podcasts are taking off, but there's so much going on in virology, I figured you guys wouldn't mind spending an hour and a half.
No, that's right.
Of course,
no, this is great.
If you didn't want to do it, you would just tell me.
Yep.
Before we start talking about viruses, can anyone guess what I need to mention first?
The listener survey. How about that?
Yeah, we want you to do the listener survey. triplemojo.com/TWiV. Please do it. So we have about 10,000 regular listeners, and we have 400 responses so far.
So that's 4%.
Yeah, we need at least 10%, right? Wouldn't that be a good number?
That would be good.
So let's try and get it to 1,000. I'll stop bothering you guys. Now, I know it's a pain to fill out, but we will not ask you to do it again, at least in this millennium. So, and it really will help us, because one result of this could be if hardly anyone responds, then we can't use advertising to support this.
Right.
And if that's the story, then so be it. But we need to know. I know it's the holidays and everyone is kind of busy, but if you have, say, 8 minutes, fill it out. You actually don't have to answer every question. You can pick and choose if you want. I know there are a lot of them. So triplemojo.com/TWiV, it would be really great if you could fill that out.
Yep, help us out here.
Yes, please.
It really will make a difference.
We don't want to pay ourselves. We just want to use the money to support things going forward.
Yeah.
All right, make TWiV even better. All right, our first story today. This is an interesting story because it is not based on a paper yet.
Right. We like to talk about peer-reviewed papers, but this is worth making an exception.
And this one...
This is based on a non-paper.
Right.
Non-paper. And so, in fact, the results we're going to be a little vague about because we're not exactly sure how they did these experiments. It's just what's been reported on. I saw this... Actually, someone emailed to me this just recently. This was from Jenny, who wrote, "Hi, Vince, and the rest of the TWiV cast. You might have come across this news already, but it would be interesting to hear the TWiV gang's take on this study done by Fouchier's group from the Netherlands. Thanks, and have a happy Thanksgiving." So she sent an article in CIDRAP, Center for Infectious Disease Research and Policy at the University of Minnesota. And it is entitled H5N1 Transmission Experiment Stirs Concern by Robert Roos. So this is an experiment done by Fouchier's group in the Netherlands. And its publication has been held up so far. Is that fair to say?
Yeah.
I believe that's the case.
By the National Science Advisory Board for Biosecurity, which is a U.S. group?
Yes.
It's a U.S. group.
Yeah. Now, it's important to note that this advisory board is they're an official, quasi-official organization, but they don't actually have a legal enforcement authority.
So how did they hold up the paper then?
They advise. They're an advisory board. They cannot say that thou shalt not publish this paper or you'll go to prison. They can just say, we really think it's a bad idea to publish this paper. And people would, if they wanted to honor this advisory board's expert opinion, the publisher would then hold the paper.
How does it get into their purview to begin with?
Let's see. One of these stories sort of went into how it came to their attention. Partly the researchers and partly the journal brought it to their attention. And this is a paper that's been wending its way toward publication for a while. That is under, I guess it's gone for peer review, and now the journal is having second thoughts about whether they should publish it. But we should talk about what allegedly the paper has done.
You don't know what journal this is, right?
I don't think that's been disclosed.
All right. So I know that when you submit to ASM, there's a box that you can check. It says, if there's any issues of biosecurity, check this box. And I have a feeling if you do that, then it goes to this NSABB.
Absolutely.
Right. So I don't know if that's what happened here. But all right, the research that they did, this concerns H5N1 influenza virus, a highly pathogenic avian influenza virus, which has killed a lot of humans, but has not spread effectively from person to person.
So this causes epidemics in birds now and then.
Right.
There has never been an H5N1 virus that has been transmissible in humans. All of the human viruses are either H1, H2, or H3, and what about 3 different neuraminidase as well? And occasionally this one, humans that are in close proximity to birds with this going on will get infected, and when humans do become symptomatic with this thing, it can be quite lethal. Like mortality rate of in excess of 50%, right? Even up to like 80%. I'm not sure, but
it's worth pointing out that some people claim that it has spread from person to person, but there is not really good evidence for that.
There have been some ambiguous cases, I think.
Peter Palazzi has said to me, and he has followed this really closely, there is really no good evidence for person-to-person transmission. But of course, if it did, this would be scary because it's quite lethal.
It's also probably been around for decades or centuries or something like that. This is not necessarily a new phenomenon.
Well, now, if bird flu did transmit person to person and retained its current lethality, that would be very scary.
Right.
Yeah.
But it's not clear that it could do both of those things.
Exactly. So in this paper, what the summary at CIDRAP says that they had introduced 5 mutations, single mutations into the viral genome. And it says here they found with as few as 5 single mutations, the virus could bind to nasal and tracheal cells of ferrets. All right, so ferrets are used as a model for influenza virus pathogenesis. And so that's kind of an obscure statement if you don't know that. I think what they have done here is change the hemagglutinin so that it will recognize a sialic acid that the H5 prefers, that is alpha-2,3 linked sialic acids. So human strains typically bind to alpha-2,6 linked sialic acids. This is the way the sugar, the terminal sugar, is linked to the next one. We'll put a picture of this in the show notes so that you can see this. The avian strains prefer alpha-2,3. And so I think they changed. And so mammals typically have more alpha-2,6 throughout their respiratory tract, and a ferret is a mammal. So I think, it's my guess, and again, we can't tell because we don't have the paper, that they changed the HA specificity so it would interact with alpha-2,6.
Right. Now, there are a couple of different pieces of work here that allegedly... It's really annoying not being able to look at the paper and the data, but the report, there's another story which is a little bit sensationalized in my opinion. I think Vincent at least would agree that, but Science did an online story. Martin Inserink, who's normally very on the ball, I think he kind of got led a little bit into this. But part of the coverage there, he did talk to some folks who have knowledge of what's in this paper. And it seems that they reported some initial work on this where they had engineered in mutations, and they talked about that at a meeting. Mm-hmm. But that was not transmissible from ferret to ferret.
Right.
But then they subsequently did this old school style and took, I guess, that modified strain probably, put it into ferrets, and then just kept the ferrets close to each other or passaged it repeatedly through ferrets to accumulate additional mutations. This is the way you would have done this experiment back before you knew about cell culture. You would have passaged it repeatedly through animals. And so they did that. They passaged it repeatedly in ferrets until they did get a variant that is transmissible or appears to be transmissible from ferret to ferret.
Now, notice we have no mention of pathogenicity here, right?
Allegedly, again, there's one line in this Enserink story that claims that it retains its lethality in ferrets even after this manipulation. But again, we don't have the data to look at.
So the 5 amino acid changes, that virus is lethal in ferrets, and then after you pass it, it's lethal and it's transmissible. Is that?
That's the implication.
That's the implication. That's what we're currently being led to believe.
You know, it's very interesting that this passage from animal to animal, which we have done for years also with different viruses, it's just an easy thing to do. Fouché calls it a really, really stupid experiment.
It's not.
I don't know why he calls it stupid.
Yeah, I don't understand that. I mean, you could call it stupid because you're making something that it's stupid to make. That's one interpretation.
I think that's probably what he meant.
But the other, you could call it a stupid experiment because it's, sort of a mindless experiment in a way. But I would disagree with that completely. I think that, this is another example of the fact that we don't really know how to change things to make them behave in a certain way genetically, but if you let nature do it or force nature to do it, you can select for things that have certain characteristics.
And I would also contest the notion that this was a stupid or careless experiment to do, that this was dangerous, because this is exactly what I might think of doing to attenuate a virus. Mm-hmm. You passage it in its something outside its normal host. This goes back to Sabin at least. You passage the virus someplace else in some other host and get it to adapt to something else, and you could very well get attenuating mutations that way. And attenuating mutations in H5N1 would be a really useful thing to have.
They seem to have forgotten that basic virology. Yes, the way you attenuate virulence by passing in different hosts. This is all just totally ignored in all these articles that are so sensationalistic. It's just all this fierce debate, seriousness, concern and Enserink, he starts his article, in the freezer here in Netherlands is a man-made flu virus that could change world history if it were ever set free. There's just zero evidence for that.
Right.
It's pure sensationalism.
It's also the other thing that bothers me is I think what happened is Enserink ended up talking to people who are directly involved with this paper, all of whom appear to be deathly afraid of what's afoot here, and are preoccupied by that. And a lot of them are also part of the what I would call the academic biodefense industrial complex. These are people who really they're very concerned about infectious agents being used as weapons, and that's kind of become their dominant theme.
Yeah.
And this NSABB is predicated on the notion of catching dangerous research. Therefore, they're going to try and find danger in the research. It's not that there's no danger here, but I think we're all probably coming toward this same opinion here. I think it's preposterous to suppress a publication for something like this.
I agree. I totally agree.
I agree.
Fouché himself is at blame, and he's quoted here as saying it's probably one of the most dangerous viruses you can make.
That to me is one of the most damning statements in this whole thing, that the author of this would say something like that.
Right. Then if you believed that, why the heck did you do this?
Right.
I think that he's looking for a little publicity here.
Sounds like it to me.
Undoubtedly.
I want to point something out about ferrets. Now, everyone will accept that a ferret is not a human. Any objection?
I make individual exemptions to that, but as a species, yes.
So, if you may remember, there is an individual quoted in this Science Insider article, Yoshihiro Kawaoka. He published a paper in 2009 where he took the pandemic H1N1 virus that had just emerged that year. He infected ferrets. It wiped them out. It ripped up their lungs. It killed them. And he proclaimed, "This is a deadly virus. Watch out." And what happened? Not a lot. It wasn't very deadly at all. Point is, ferrets really don't tell you everything that you need to know about the virulence of influenza viruses.
Right.
So I would say that this is meaningless in a ferret. And to make proclamations that it's dangerous is completely inappropriate in an animal model.
Yeah, and I think this Enserink story quotes one of these people as saying that the ferrets, ferrets aren't human, but in studies to date, any influenza strain that's been able to pass among ferrets has been transmissible in humans. And they make the implication that ferrets are a good predictor of pathogenesis in humans. But as you just pointed out, the data argue otherwise.
Yeah, it's just not true what he's saying.
So mice lie, monkeys exaggerate, and ferrets are weasels?
Yes.
Good episode title.
I like that. Except we haven't involved mice and monkeys in this yet. But
it's a nice continuation. The broader issue here, which I'd like our thoughts on, is this whole idea, and I guess you have already touched on it, Alan, this whole idea that you shouldn't do certain kinds of research, and if you do, you shouldn't publish it. What do we think about that?
This is, in the computer world, there's this, there was a number of years ago a raging debate about the best approach to security. Do you keep a computer system secure in by taking the approach of keeping everything secret? This is the Microsoft approach. Which kind of tells you how well it worked out as a security strategy. But the idea is you keep your source code secret, you keep as many secrets as possible about how the system works, and that makes it harder for hackers to break into it. The other side of that is that you get security through transparency, that you open the source code to your operating system and everything else, and you say, hey, everybody, here's how this all works. You want to try and hack it? Go for it.
Yeah.
And that is the Linux Unix approach. And there's no question at this point which of these strategies works better. Because if you have this security through obscurity strategy where you try to keep everything secret, first of all, you can't. Secondly, it's when the lights are off, things fester. And you'll get just the people who are inside an organization working on your computer code, and they're aware of potential flaws, but they can't say anything about them. and you're just waiting for some hacker with or without inside knowledge to exploit one of these flaws. Whereas if you have everything out in the open, then there are no secrets, there's nothing to exploit, and anything that is exploitable, probably the good guys who outnumber the bad guys are going to point it out to you and say, hey, somebody could exploit this. And I think we're now having the same discussion about biology, about infectious disease. Is it better to try to keep this stuff secret, the Fort Detrick approach, or is it better to do the research, find out as much as we can about the biology of these potentially deadly viruses, and publish it promptly in peer-reviewed journals so we can all look at it and say, hey, these mutations make this virus more transmissible in another host species. Let's look at that. Let's develop maybe antivirals that target whatever mechanisms it's using. Let's use this as a vaccine production system. Let's explore that possibility, because somebody's going to eventually, either humans or nature.
Yeah, I would go. I agree. I would go with the transparency approach. I must say, it's interesting because with this, what I find is with this story sort of half out there, I want to know what the data are.
Sure,
that's one of my predominant feelings about this whole thing is I want to see the data.
Yeah. See, the problem, as Alan has said, if you restrict the data, that means you think you know who could solve the problem.
Right.
And I don't think you can predict. It could be some guy, some obscure virologist somewhere looks at it and goes, oh, I know how to deal with this, and he or she does it.
Right.
So more people that look at it, the better off you are.
Yeah, and the good guys outnumber the bad guys in this case, again, and have much bigger resources.
Yeah.
So is some terrorist group going to regenerate this virus and turn it into the next plague? Well, I don't know, but I know that the world would be a safer place if we all knew what this thing looked like.
I mean, that brings us to this issue of, don't we think, we've said this before, nature is a much tougher incubator than the laboratory.
Absolutely.
The hubris of this is absolutely astonishing to me, that we think we can make a deadlier flu virus than nature already has.
Really?
We already got 1918 flu. Sequence is published.
Yep.
That's one thing.
Anybody with the capability of doing anything like this to try and make it nasty or whatever would be a shorter route to just recreate the 1918 flu. We got smallpox as well, and if anybody thinks that's child's play, they need to just go look at a few pictures. You can, with the smallpox sequence is published. You could theoretically put that back together. The nasty bugs are already out there. Nature can create nastier ones.
Yeah, so it's all been ignored in these articles that we have all these viral sequences, 1918, smallpox. They're out there. Everybody looks at them. Nobody's made them. No one's deposited them anywhere. And this is something made by a person. You make a couple of mutations and a couple of passes. There's just no way that trumps what happens in nature.
Well, it could. We don't know whether it does or doesn't, because the experiment hasn't been published and hasn't been explored. But the idea that the assumption that our prediction in ferrets is going to play out in humans, and that we've beaten nature to producing a deadlier flu virus, I think that's really out there.
I understand that, but I just feel anything we make and put out there is going to be homogenized by nature eventually. Just like you put the virus in rabbits in Australia. After a year, it's everybody's evolved, and
of course it killed 99% of the rabbits in the meantime. But
yeah, but that was a naturally occurring virus. It wasn't a human-made virus.
That's right. True.
Yeah. So I think this is. I mean, I'm sorry we don't have this paper. I guess when it comes out, we'll take a quick look at it again to see if there's anything new in it.
So another thing that's worth, at least a couple of comments on here is this process, the review of this kind of stuff. Okay, so I think it is, let's say it's not inappropriate for scientists to question the possible utility of this stuff, to look into the possible dangers of stuff that they do, and have a reasoned debate about that. I think that that's important. And you can question how that should be done. We have this device that's the NSABB, which certainly they have a mission, but they also have a membership that contains a lot of very reasoned scientists. And I would much rather that they be looking at this stuff and thinking about it than it go over to have, Congress trying to legislate this. And in fact, I had an early encounter with this board when it was first constituted. I don't know if it was when it was first constituted, but fairly early on, when Lynn Enquist was they were looking for models of how to identify research that could potentially have dual use. And I don't know if you were involved with this, Vincent, but he sent out a sort of an instrument to editors asking, with some sample papers that are possible dual-use papers, and a questionnaire that might accompany papers like that, to try and determine whether or not, according to the editorship, they would be dual-use things. And I had a fairly negative reaction to the whole thing, and that got me involved with discussions about this board. At any rate, one of the things that Lynn said is, look, we need to do this because if we don't do this, Congress is going to do it, and you don't want that, okay?
Yeah.
Not that Congress is necessarily a bad thing all the time, but it's not necessarily the most scientific approach to something like this. This panel has good scientists on it.
I think self-regulation is great. Asilomar,
yes.
Right, is the model.
Absolutely.
But I don't know about this panel. Everybody seems gung-ho on saying this shouldn't have been done. We're going to issue additional recommendations about this kind of research. I think
they've set them up so that the purpose of this panel is to say no.
Yeah,
because if they never do, then they don't have a purpose to exist. My main problem with this is not that the discussion is taking place. I would agree that there probably are experiments that should not be done, and should not be. There might be data that you just don't want to exist out there, but that's exactly the issue. Is that these things need to be discussed before the work is done. And people, if you really think that this is a stupid experiment, you shouldn't do it. If you sincerely believe that you're making the deadliest virus in the world, then that was a bad experiment to do, and you shouldn't have done it, and it shouldn't have gotten to the point where we're debating whether to publish it. But my feeling is once the experiment's been done, and particularly when part of the data have been presented at an international conference of some kind, and the rest have been sent out to peer reviewers, which is not a top secret clearance type of situation. This is something that went out to scientists who are the worst people in the world at keeping secrets. They may have shown the papers to colleagues while they were reviewing them. It's been submitted to this board. You've got probably hundreds of people who have access to this in one way or another. It's not secret anymore. And so to sit there and say, oh, it shouldn't be published, well, the horse already left. Closing the barn door now is not really going to help.
Nope, absolutely not. But I don't think this is an experiment that shouldn't have been done.
No,
I think this experiment.
I think this was actually a really good experiment and should be published.
As a guy from Rutgers, Richard Ebright, a strong interest in biosecurity issues, says this work should never have been done. I don't know why he's saying that.
Right. Oh, right. What I'm saying is there is some work that probably shouldn't be done, but I don't think this is it.
This is not it, but this is being used now as a litmus test.
Right.
And the public is going to get scared about this because they're not going to really understand all this nuance. And they're going to just see this statement of Fouchier in this article, which says it's wrong to say that only H1, H2, and H3 viruses can cause pandemics. He says his study shows all of that to be wrong. It shows no such thing.
Not true.
No such thing.
But it's
unless
going to be
he
quoted.
has human data.
Yeah, but it's going to, that's going to be pulled out of context and quoted.
Right.
So this is really annoying. Well, maybe people will hear us.
Maybe.
This reminds me a little bit of when Ecke Wimmer made polio, from scratch. It's the same sort of blow up in the media,
Well, that was even worse. Really, that was even worse because it had already been done 20 years before.
Right. Crazy.
Yeah. Well, that still is used as an example of making pathogens,
Right. So there are a lot of people on this committee that are not quoted in this article that I respect as very reasoned scientists, and I'm glad they're on this committee and dealing with this. It'll be interesting to see what happens.
We can find the list of people on this.
Yeah,
committee.
If you look down, it's a matter of fact, it's at the list of references on the bottom of the SIDRAP thing. Takes you to the NSABB, and there's a somewhere in there's a link to the.
Yeah, got it.
Yeah, you've got the voting members and ex officio.
Oh,
members.
Look at this! Our buddy Mike Imperiali, who we just saw.
Yep. Ken Burns, Stan Lemon,
Stu Levy, who was on, and Arturo Casadeval, who were both on TWiV.
Jeff Miller, Lynn Enquist.
Yeah.
So there's a lot of good people.
So we didn't hear them. David Relman.
That's right.
We did not hear the reasoned voices. Claire Liggett. We only heard the dangerous thing. Oh, okay.
And it's possible that many people on this committee declined comment.
Yeah, sure.
Right.
Sure.
I mean, I gave Enserink a little bit of grief already, but it's entirely possible when you're a reporter working on a controversial story like this, you call people up and they say, "I'm not discussing it." And then you end up with the people who have some strong opinion, and it may all cut in the same direction, and that's possible that's what led to this story.
And the majority of the people on this committee are card-carrying scientists. There's a couple of legal types, and there's a couple of military types, but most of them are quite legitimate scientists.
Yep. I will put a link to the NSABB so you can check that out.
So I favor a discussion of these sorts of things ahead of time if you're really concerned, but this paper is not that type of situation in any case.
And I thought ultimately that the solution that the Journal of Virology came up with, rather than some weighty instrument to try and judge whether or not something deserved additional attention, of just having a little checkbox made sense. Though, as you pointed out, Alan, that's kind of closing the barn door after the horse is already out,
Yeah.
Nevertheless, that's in terms of the journal's approach, that's a reasonable way to approach this. I don't know that other journals, they must have some sort of a process, but certainly it's not that.
No, I think this is something that should be approached from the funding agency side. Either as people are writing grant proposals or in a general orientation, you say, hey, give some thought to this. There are bad people in the world who might want to do bad things with biological agents. If you're working with something that might fall into one of these categories, just keep it in mind. And if you are trying to make a more virulent strain of something, give it some thought. And if you're concerned, here's this board you can go to and take your concerns up with them before you do the experiments.
Actually, something our listeners might like to know is that most, if not all, institutions have some sort of biosafety committee.
Yes.
That reviews all kinds of research. There are guidelines for these committees, but there's also considerable variability, I think, from institution to institution. I can tell you that the University of Florida has a pretty conservative biosafety committee, and I would imagine that if I had proposed these experiments to them, they would just laugh me right out of the place. But, as I say, there's but the point is that there are mechanisms in place in most, if not all, institutions to preview this kind of stuff. They even in the SIDRAP article, I think, talk about some European or a Scandinavian committee that had to vet this particular research before it was done and gave them the go-ahead.
Yeah. And if I were on a committee like that, I certainly would have given this the go-ahead. It's a very good experiment to do to try and learn more about a virus that could become dangerous in the future all on its own.
I believe the composition of these biohazard committees is a public record. Isn't that correct?
Yes, generally.
Right, so then I can say that.
Well, I got this right off the internet, right?
Yeah, I can I am on the Biosafety Committee for Columbia, and I would review this kind of experiment. And we have many dangerous experiments that we review. We just make sure that the containment is appropriate.
Mm-hmm.
Right.
We don't worry about giving people the wrong information. It's really not our purview. Our purview is institutional biohazard committees are to keep it safe for the community.
Right.
And we don't talk about publication. But I thought if this came past my committee, we would say as long as you're doing it with the right containment, go ahead.
Yeah.
I mean, let's say Ian Lipkin came to us and wanted to do an experiment, and we said no because we don't want a terrorist to get the results. What do you think he would do with that?
He'd go to another institution.
Yeah, so it's not what our purview is. We have to just keep it safe. Anything else on this before we move on?
No, I think that's a good bottom line, is that it should be if the work is safe for the researchers in the community that they're in, and the Biosafety Committee has judged it that way, then the issue of publication should be, well, hey, the experiment was done. It ought to be published.
Yeah, and I think also Martin could have done a different job writing it, not as sensationally.
Maybe. It depends, as I say, it depends on what he was able to get a hold of. I think sometimes these things go this way, but it's also kind of alluring to pursue a story that sounds sexier.
I don't know. I think you would have written it differently with the same information, but we'll see.
I don't know if Martin's trained as a virologist, though.
Right.
Actually, there's somebody on this Michael Osterholm is a member of this committee, and he's quoted in this article as saying the experiments have the full support of the influenza committee, the influenza community.
Good.
Yeah.
They should, as they should. I had emailed Peter Palese just before we started about this, but he hasn't responded yet. Nope, he's still eating his turkey, I guess. So next time. He always has an interesting view. All right, let's move on to our other story, which happens to be also about influenza virus and perhaps a way to treat infections that doesn't involve antivirals or vaccines. This is a paper in Nature Chemical Biology, which is entitled Chemical Inhibition of RNA Viruses Reveals REDD1, which is a protein we'll talk about, as a host defense factor. The first author is Miguel Mata, and the last is Beatriz Fontura. And this is from Texas, the University of Texas Southwestern in Dallas, Mount Sinai.
And also Mount Sinai.
Mount Sinai, Adolfo Garcia Sastre, and a bunch of people in between. This deals with the NS1 protein of influenza virus. Non-structural protein 1. This is actually a tough paper. We're going to have to work hard to make it clear to everybody.
Right.
NS1 is a very interesting virulence factor of influenza virus. It is, if you take away this protein, the virus will replicate, but it is severely compromised. It's severely attenuated, and it will only cause disease in animals that lack immune systems, lack innate or interferon immune systems. It's involved in antagonizing a lot of host processes, export of mRNAs from the nucleus, signaling leading to interferon production, and so forth. So they decided to look for small molecules, small chemicals, that would reverse this NS1 inhibition of host functions.
Right. So NS1 is sort of a jamming device for the virus.
That's right.
It's gumming up things that would otherwise alert the immune system that, hey, this is a virally infected cell.
Yeah, big time.
And it is there apparently solely for the purpose of confounding the immune response, right? In one fashion or another.
Yeah, a deletion, a virus lacking it will grow in cells.
Right.
It's remarkable.
Interesting.
Yeah. It's very interesting. And it's got a lot of functions that we don't even know about, as this paper will reveal. So they did a high-throughput screen. That is, you have a library of 200,000 synthetic compounds, which are easy to come by these days. You can buy them. And their assay was they put into cells NS1 on a plasmid, and they have on a separate plasmid a gene encoding luciferase, which will give them something they can measure. They can measure light. All right, and when you do this, put NS1 and luciferase together in a cell. NS1 inhibits the luciferase production.
Right. So they have this library of compounds. This has been a strategy in the drug industry for a number of years. You take this library of compounds that you think you might be able to turn into drugs, and you screen for some outcome. And in this case, they're screening for these compounds' ability to inhibit the activity of NS1.
All right, and this is all done robotically with automated machines and.
Right, 200,000 compounds. You don't want to be doing that with a pipetter in your hand.
So I think we once mentioned, maybe it was on another show, how many compounds a person could do in a day. Does anybody remember what that was? One person with a pipette, I think 100 compounds a day.
Something like that.
Yeah, that's a lot of days to get through 200,000.
Yeah, and they do these they do these assays in a super miniaturized form. I think here they use 384 well plates.
Yeah.
In industry, they've now moved on to 1,536 well plates.
Microfluidics
Devices sometimes, although in this case you just have wells on a plate. For people who haven't worked in a lab, if you imagine an egg carton and you shrink it down and put more and more and more little divots in it for the eggs, that's what one of these multi-well plates looks like. It's all these little tiny wells arrayed out in a grid, and this is all, the fluids are added and moved from one to another, all by robotic pipetting devices.
Now, interestingly here, what they did is use a very low concentration, 5 micromolar, because you want to pick up things that are active at low concentrations. You don't want to have to use a lot of it. Right?
Yeah. If you need grams per milliliter, it's not a drug.
So they look for compounds that inhibit the NS1 function, and then they took the positives, and then screened them for preventing cell killing caused by the virus.
Right.
So like a double screen. Yeah, you do 200,000, you come up with X compounds that inhibit NS1, then you take those and screen them again in infected cells.
Right.
So they came up with these, what's the damn name? Naphthalamides, which is dihydroxy chicken wire.
Yes.
As Dixon would say. These multi-ring compounds, which are very popular here in chemical biology. And they made some modifications to improve the half-life of their main hit, and they came up with something that lasts longer. And
in the process of doing the modification chemistry, came up with a derivative that also didn't work at all that they can use as a negative control, which is good.
Right.
So you notice it's very interesting here. So these are compounds 1, 2, and 3. 1 is inactive. 1 is the original, 2 is the inactive one, and then 3 is more potent.
Right.
Every time they mention this in the paper, it's a hyperlink. Did you notice that?
Yes.
They've got this thing, the Nature Chemical Biology is this that they highlight all of the compounds and all of the genes and proteins.
So you can click on it and go somewhere and get more information.
Right.
Except when I click on a bunch of these, I get the DOI handle you have entered is invalid.
Great.
Oh, great.
But they're trying.
Yeah. So then they take this drug, and the question is, what is it doing? And they do a lot of experiments. They show that it inhibits viral replication. It keeps cells alive.
And they also find that it inhibits not only flu virus replication, but vesicular stomatitis virus replication.
Right, which would then...
Right, that's an important control, because that tells you that's what tells them that it's messing with the cells, not with the virus.
Now, one of the things NS1 does is to inhibit the transport of messenger RNAs, which are made in the nucleus, inhibit their transport out into the cytoplasm. And that's one of the reasons why NS1 has a strong effect on the cell, because all the cell messages can't get out, so they can't be translated in the cytoplasm. And this drug partially reverses that inhibition.
The immunofluorescence pictures that show that are striking.
Yeah, nice. They're very nice.
I didn't realize how strong the effects of NS1 are.
Yeah,
so
nice.
They got this thing where they actually probe for polyadenylated RNA, so they're looking for messenger or pre-messenger RNA in control cells, and it's everywhere. If you infect with flu It just lights up the nucleus. Everything's jammed up in the nucleus.
That's nice.
And then if you add this compound, you get a sort of a mixed result. Some cells look like the RNA is distributed uniformly, and some it's still stuck in the nucleus. It's very striking.
Yeah.
So they want to figure out how this compound works. It looks like it might be targeting some cell activities, especially the fact that it would block VSV would suggest that it could be a cell protein or a cell function. So what they do is another screen. This time they add this compound to cells and they look for what genes are expressed with or without it. They do a gene expression profile with or without compound. There's another big data set that's published here. And what they find is that there is a very particular pathway in the host that this drug, this compound, number 3, seems to be affecting. And we have here another wonderful acronym, the mTORC1 pathway. Mammalian target of rapamycin complex number 1.
Right.
And there are 2 of these complexes, TORC1 and 2, originally defined by this immunosuppressive drug rapamycin, which have amazing roles. These TORCs have amazing roles in homeostasis. They're involved in all kinds of things, sensing stress, sensing nutrient deprivation, and then they're sort of central players in regulating how those sensing events are occurred. So if you have stress, you want to shut down translation. So these TORC proteins are involved in that. So that is where this compound is working on, this TORC pathway.
I have to say that I found, for me at least, the microarray analysis in this paper really impressive, because usually when I see a microarray analysis, and what this means is that you basically take the RNA and you can hybridize it to a chip that has representation of sequences from every gene in the genome and ask what RNAs are there. And usually you get out just this incredibly huge data set that you don't know what to do with. It's too much data. Now there's a lot of bioinformatics that have been developed, one of which is to analyze patterns in these RNAs, and you can see that certain sets of genes that are turned on represent a certain pathway. And in this particular case, that turned up this TORC pathway as being really stimulated. And I was impressed with the fact that the microarray analysis in this case really fingered a very specific pathway and gave them a very specific answer.
As opposed to 100 different
As
pathways,
opposed to just a whole bunch of genes turning on, and you say, oh, well, let's study this one. This really led to an answer.
Right.
So they identify a specific protein. That is REDD1 from the title, and that stands for Regulated in Development and DNA Damage Response. It was a protein described a number of years ago, given that name, and it's now known to be part of this TORC pathway. All right, it is actually an inhibitor of TOR, and we'll see how that plays out in a moment. So REDD1 is induced by this number 3 compound. All right, so you add this 3 compound to cells, it induces REDD1. Their microarray shows that very clearly. Mm-hmm. And you need to have REDD1 in cells. This is a protein. It's got to be there for 3 to work, compound 3 to work. So they did some cool experiments where they got cells lacking the REDD gene, and in those cells, 3 has no antiviral effect.
Right.
So you have to have REDD1. And it works, as we said, not only against influenza, but VSV as well. And it turns out that REDD1 is initially induced by viral infection. It's an antiviral, part of an antiviral pathway. It's induced, and it will shut down this mTORC complex. Now, the mTORC complex is involved in stimulating translation by a variety of phosphorylation events. And when a virus infects a cell, that initiates a stress response. The cells shut down their translation in part through this mTORC pathway, and it's in part through REDD that shutdown occurs. So viruses infect cells, REDD goes up, but flu then turns REDD off. So translation can keep going. It can make its viral proteins and replicate its genomes. But what this drug does is to keep REDD being made. It keeps its synthesis constitutive in cells infected with the virus. So instead of the virus shutting it down, you have number 3 around and you make REDD, and therefore translation proceeds.
Right.
This is one of these really complex, multiple negative type things that drives me crazy. There's this chain of events that's really hard for me to balance in my head.
Right. It's not inhibited.
Right. So flu wants, anthropomorphizing it. Flu wants TORC to be on. Right?
That's right. It wants TORC to be on so that protein translation proceeds. Right.
So the antiviral response is
to turn TORC off.
Right.
Is to turn TORC off by turning
on REDD.
By turning on REDD. Right. But then the virus basically ordinarily turns REDD off.
That's right.
But the drug turns REDD back on.
Yeah.
In order to allow TORC to be on.
That's right.
But then the drug keeps REDD or induces more REDD synthesis, which turns TORC off, and the virus doesn't like that. My God.
It's incredible. So
this
is a new function for REDD. This is totally revealed by this inhibitor, so it's quite good. Even if this drug never makes it anywhere, it's for research purposes, it's revealed a great deal about a new antiviral protein.
Yeah, and actually high-throughput screening has not panned out terribly well in the drug industry because it's been used mostly as a way to try to find new drugs, and it hasn't really generated as many as its cost would seem to require. But there's been an argument for a while that, well, maybe we can use high-throughput screening as a way to get at mechanisms and then more intelligently design drugs that target those mechanisms. And this is going down exactly that pathway, and it suggests that could be a very promising approach.
Yeah, whether or not it pans out, who knows? It depends on many variables.
But this is very encouraging because by industry standards, 200,000 compounds is not a big library.
No.
Right.
So this is a relatively small automated screen, and bang, they've already found a whole new pathway of viral inhibition that you could potentially target to go after a nasty virus. A couple of nasty viruses, maybe even more.
Okay, so to summarize it, we have this new compound, number 3. You put it in cells. It keeps the synthesis of REDD1 going when flu infects, and that REDD1 prevents viral infection by shutting down translation through this mTORC complex.
Right. Now, just to make this clear, you can't go out and ask your doctor about 3.
Yeah.
Can I
have some 3?
Yeah. You may not want any, as a matter of fact.
There are no animal studies here, so we have no idea. I mean, this is messing around with a very central cellular signaling pathway.
Yep.
So we have no idea what the side effects might be. There are no animal studies. For all we know, the minute you get it out of culture and into animals, it'll turn out to be extremely toxic and worthless in therapy.
And it's worth noting that here we have this pathway that apparently is important enough that 2 unrelated viruses have thought to shut it down.
Mm-hmm.
Right.
And yet, evolutionarily, we haven't decided to turn it back on again. Maybe there's a reason for that. You know, we might be at the point where you don't want to just upregulate REDD because, as you say, Rich, it could have some horrible side effect. Or maybe we've outsmarted the system and we can get a leg up on it.
Well, this is a good example of an approach to antivirals that a number of people are pursuing, which is trying to find cellular targets rather than viral targets, okay? Because the problem with viral targets is that the virus can mutate to get around whatever drug that you are using. And it might be theoretically at least, more difficult. It's not going to happen as frequently probably almost never, that an individual human is going to mutate to resistance to the drug. Okay?
Right.
I want to read a paragraph from the discussion which I think summarizes this whole approach really nicely. They write, there are essentially two approaches for identifying unique host processes that are involved in antiviral functions and can be exploited therapeutically. One is to learn as much as possible about the host mechanisms required by the virus and then test the effects of inhibitors on them. So that's what many labs do. They identify what host proteins you need for your particular virus, and then they eliminate those proteins by RNAi and then ask what's the effect on virus replication. The other, which they continue, is to take an unbiased approach and screen for chemical inhibitors of virus functions or host genes required by the virus. So in the way they set up this screen, they could have identified inhibitors of NS1, right? Because that's where the readout was. So I think they were lucky that they got a host protein here. It could have gone either way.
Right.
Anyway, this got some press, so I thought it's a really interesting paper, really nice.
Right. Whether or not it turns out to be of value therapeutically, what we learn about the replication of the virus from this is really important.
Yeah. We have a new host defense factor, REDD1.
Yeah, and how many other viruses is this involved with? You've got 2 in this paper. It's worth going and looking. So virologists, go out, see if REDD1 is involved.
Okay.
I'm really impressed with this one figure early on where they show cultured cells and they infect with flu, and there's a really dramatic cytopathic effect. The cells all look terrible. Yes. And then they treat with the drug and the cells just look normal. They just shrug it all off. Yeah, that tells you two things simultaneously. One, it's not killing the cells. And 2, it's preventing the virus replication.
Yep.
I spend a lot of time looking at infected cells, and when I see something that prevents a cytopathic effect like that, it really catches my attention. That's pretty impressive.
Yeah.
So they have this cell line of cells that lack the REDD1 gene, and in their case, flu replicates better in those. So anyone could get these cells and test their virus in them and see if it does any better in those cells. So that's an easy way. You don't have to even have the compound. You can go right to Red October.
Yeah.
All right. Very good. REDD1. And let's move on to some email.
Yeah.
Just before we had Jeremy on, he had sent an email, and I forgot to bring this up with him, but he wrote, "Thought you might want to know about Uta von Schwedler. She was a postdoc with Didier Trono in San Diego, then with Wes Sundquist in Salt Lake. Then she worked with ex-Colombian Ela Singh. She has some very important papers in our field that you might call classics. I refer to them again and again. Papers defining the VIF phenotype in HIV-1, capsid structure, function, and escort." And Uta recently passed away. We have an obituary photo and article from the Salt Lake newspaper, which we'll post. Jeremy writes, "I knew Uta well from Cold Spring Harbor. She attended frequently. She once took my daughter swimming at Cold Spring Harbor. When I showed the obituary picture to my daughter, she pointed out something ridiculous that I am embarrassed to say I never noticed. Uta lived in Utah." She was a subset of Utah. And then Wes Sundquist wrote, "Jeremy, thank you for compiling that information. As you say, Uta had a major impact on our field and touched many people in addition to her scientific contributions. A Facebook page has been set up in honor of her, and we'll provide the link for that as well. This summer, one of her papers was selected as one of the 30 most influential papers in 30 years of HIV research, and Wes wrote a commentary on it, which we'll link to as well. The selection occurred this past summer, and I'm pleased that Uta knew about the selection and saw the commentary before she passed away. You're right, Jeremy. Uta did live in Utah. She would also sometimes take the light rail or bus systems, which are run here by the Utah Transit Authority, UTA.
Huh.
It used to amuse us that over the years we'd sometimes see her sitting at a bus stop bench labeled UTA or see billboards that said things like, "Go UTA." That's pretty good. So I thought we should just mention Uta because she apparently was important in the HIV field, and she passed away. Let's see when. I have the...
Not too long ago.
I have the obituary right here. Uta von Schwedler, October of this year. So the newspaper is the Salt Lake Tribune, October 12th, 2011, 1962 to 2011. We'll post that picture. Very unfortunate, but a good contributor to the virology world. Thanks, guys. All right, the next one is from Matt Evans, who's been on TWiV a few times, and he sent an article, Pox Parties in the Age of Facebook. And he said, what do you think of this? Giving out lollipops infected with chickenpox virus.
I saw this a while ago.
The irony of this is just completely off the scale.
Yeah.
People who object to giving their kids a proven safe vaccine against this potentially very dangerous virus were apparently wanting to give chicken pox to their kids directly from infected lollipops.
Yeah.
It's just...
I know. Reminds me of variolation.
Exactly. You're going to expose the kid to this nasty, nasty virus, and that's your vaccination strategy. And it's a vaccination strategy, but it's a stupid one. And there's a perfectly good vaccine against this virus that is available, and people just can't understand that.
Yeah, it's rich, right? They used to give smallpox to people, and a bunch of them would die, but the rest would be protected, right?
Right.
And we developed vaccines to get around that, right?
Right. Yeah, so as Alan says, this is really unfortunately misguided. It's a really sad example of the degree to which the public is distrustful of science,
right,
and misinformed about what goes on.
Totally crazy. Absolutely crazy.
It's also been pointed out here. It's pointed out in this article. I've seen some of this as a follow-up, that the mailing around these slobbery lollipops and stuff, which is one of the things that's suggested, is illegal. You can get thrown in jail for this, apparently, or subject to
mailing an infectious disease. Yeah.
Right. Oh, well.
Yeah, I thought we should mention that because it's really bad. It is really, really not smart. The next one is from Anonymous. I'm writing to tell you how thankful I am for the work you are doing with your brilliant podcast. I'm a bachelor in biochemistry, soon to start a molecular microbiology master's program. I would like to share with you the last 4 years of my life. In late August 27, I was traveling in Southeast Asia during my summer vacation. I visited Indonesia and Malaysia. Unfortunately, I caught a tropical virus, namely chikungunya. I'm sure you're familiar with it. In rare cases, the virus gives you chronic joint pains. I'm one of these rare cases. Over the years, I have consulted numerous doctors. Recently, I consulted an anesthesiologist who provided me with a painkiller, which has changed my life. Over the past 4 weeks, I have walked more than 200 miles. It's hard to describe how joyful I am. For every minute I walked, I was listening to your podcasts. They really helped me get my fascination for microbiology back. In 4 years, you forget a lot. Your passion, your drive, it takes a serious blow. You have helped me find this long-lost companion, and for this I'm very grateful. So I'm sending you my deepest thanks. I hope you'll keep up the good work. For my part, I'm looking forward to the next many miles in your company. P.S. Did you ever do a part about chikungunya?
I don't know that we ever did a whole episode, but I looked it up. We at least mentioned it and give it some coverage on TWiV 37 and TWiV 69. So have a look at those.
Yeah, I'm sure we'll check on it again at some point. All right, we have an email from Danielle, who is from the University of California, Santa Cruz. She's an undergrad majoring in molecular, cellular, and developmental biology, bioinformatics.
My alma mater.
Is that right?
My undergraduate alma mater.
Okay.
Go banana slugs, right?
Go slugs. She should go say hello to Harry Noller. And he's the guy who got me my start.
Oh, yeah. She's got to go say hi to Harry. But he might scare her.
He's not so scary, you know.
Does he still have that big beard?
Well, it's not as big. He's not nearly as scary as he used to be.
Okay. Anyway, Danielle basically writes about the Microbe World app. She was looking for an app to stream the shows to her iPhone or Android device, and she said it seemed like there was one review, and it said it was broken, and you should make your own someday. She said, I love to listen to podcasts as I do lab work, so I subscribed to your podcast with Google Reader, and now I can listen on my Android phone, and it also works with the iPod and iPhone, so you should convince a computer science undergrad to make an app. I want to thank you guys for doing this podcast. As an undergrad, I'm not really sure what I want to do post-grad, and I've always been interested in virology. My college has Phil Berman. He worked on one of the HIV vaccines, teaching vaccine-related classes. We don't really have a virology department, and I wasn't sure what a virology lab would really do or look like. However, because of your podcast, I'm pretty sold on doing something virus-related in the future. And our friend Chris Condian at ASM responded. I sent him her email, and he said that the app will work, but you have to have an internet or 3G connection, or you can't play the podcasts. And he says, basically, if you like it, write a review. There are also some alternatives to the Microbe World app. It is $4.99, which goes to the American Society for Microbiology to support their activities. But Stitcher Radio will also stream the podcasts to your iPhone or Android device, and it's free. And there are other solutions as well. But the Microbe World app supports ASM, so check that out.
So Chris also says that apparently there's only this one negative review that Chris figures was because the person wasn't connected to the internet. And he says he's tried to respond to the review using the comment review fields to sort of counteract the damage. But since he's a content creator, he can't do that. So maybe, who is it, Danielle? Or,
right, Danielle.
Maybe she ought to go on there and comment back. See, make it work. Go on, comment back, and neutralize that problem.
Yeah, or someone can do it.
Someone.
Okay, Alan, can you take the Simon email?
Sure. Simon says, "Hi, Professor Racaniello. I'm delighted to hear someone of your caliber has joined the CFID Scientific Advisory Board. CFS research needs more high-grade input. I'm also glad you met some patients and discovered that most of us are reasonable people trying to cope with a very difficult disease, and usually our illness kicked off after an infectious episode. I've enjoyed your podcasts on XMRV, and I'm sorry about the amount of flack that appeared on your site. A tiny but vocal and vitriolic minority of patients give the rest of us a bad name. Yes, we try to argue with them on forums, but you can only be torched so many times before you realize it's not only deeply unpleasant, but also pointless. Anyway, thank you for your commitment to trying to solve CFS. It is appreciated." Simon's in the UK.
All right, Rich, can you take the next one? And
right, skip over the TWiP part.
Yeah, this is from Trudy.
Okay. Trudy writes, "Now, about TWiV, can you cover herpes B? I actually knew a patient who sustained a bite from an infected monkey quite a few years ago. So this is a monkey virus that can get into humans, and if I'm not mistaken, it can be quite lethal in humans. Am I thinking about the right agent?
Yeah, that's right. You got it.
The patient was a vet and was treated with a series of IV intravenous acyclovir infusions. Acyclovir is a drug that's active against a number of herpes viruses. She was still living as of a couple of years ago when the doc who treated her lost track of her, but it was probable that she was still okay. The treatment was experimental then. Also got a huge kick out of your comments about D.A. Henderson's remark about the polio vaccine. I had the pleasure of meeting him when he came to the College of Public Health at the University of South Florida to talk about weapons of mass destruction. We spoke about vaccine-preventable disease and the growing disconnect with those who are anti-vac wackaloons. Since I was involved with outbreaks of assorted diseases, including measles, we had quite a discussion. So many docs today have no idea what the rash illnesses look like, and there is pressure for the newest grads to adopt CAM. CAM?
Complementary and alternative medicine.
Woo-woo. Woo-woo.
Sorry, it's...
It's okay. It is of great concern. My mother-in-law had polio when she was a young mother. She suffers from post-polio syndrome now. I absolutely love your podcast. Thank you for all your hard work between your podcasts and ID Doc, Mark Crislip's Quackcast, Pusscast, and Persifliger's Compendium of Infectious Diseases. I am in hog heaven, tapeworm-free. Holy cow. I eat, breathe, and dream about tiny animalcules. No need to respond to this long-winded tome. Just had to write. Miss working in public health. Nice letter.
Yeah.
Yeah, the rest of it is on either has been or will be on TWiP soon. All right. That does it for today's email.
We ought to say something about herpes B sometime.
Oh, yes. That's a great idea. That's
a good idea.
I just added it to the list of topics.
Good.
We'll do that. All right. Let's do some picks of the week and start with Rich.
Okay. So this may turn out to be one of these things that you guys and all the listeners already know about, and I'm like totally behind the times. But nevertheless,
I had never heard of this.
Good. I'm glad to hear that. While I was in Michigan, which, by the way, was absolutely a delightful visit.
Oh, yes.
I had such a good time there, and it's such a nice department. I was really impressed. I really had a great time. So I was sitting next to Kathy Spindler while you were giving your talk, Vincent, and she was taking notes with this thing called a live scribe, which is a recording pen. So she was taking notes, and you do it on special paper with this pen. The pen records the talk, so the voice is actually recorded as you're taking notes. And then when you're done, when you go back to your notes, anywhere you touch in the notes, it goes back to the place in the talk when you were taking notes and plays it back. Okay, so you can see what you were talking about. And it remembers everything you wrote. And it's even got little things on the top and the bottom of the page where you can search and rewind parts of the talk and all that kind of. It's amazing. And then you can transfer the whole thing to your computer if you want. So you have a digital version of the same thing. But I've had this experience. Well, I don't take notes anymore because I just can't do it anymore. But I've had this. I remember taking notes. And looking back at my notes later and thinking, what was I thinking? What was actually going on there? And the Live Scribe allows you to go back and replay that part of the talk. So it's pretty cool.
That's awesome.
You're going to get one for Christmas?
No, like I said, I don't take notes anymore.
You don't take notes. Yeah, it's for people who do, right? Right. I still take notes, as you saw. You were surprised.
I take notes. A few years ago, I finally switched to taking notes on a computer because I finally found a workflow that could accomplish almost what paper accomplishes.
Yeah, I was thinking about that because I was thinking a lot of people probably going over to the computer. One of the ways they advertise this Live Scribe is they show somebody taking notes, and among other things, they've doodled a little picture. That would be hard to do on the computer. Okay, so
yes,
there are still some things you can do with a pen that might be difficult to do with a computer.
Yes, that's one thing that I can't do in my computer notes. But since I work mostly in the world of text, it's not too much of a problem.
Rich, at your talk in Michigan, my host Stefan was taking notes on an iPad. So he had a special stylus that you can use, and he had a program. I forgot which one. There are a number that you can use to write. Handwriting. It doesn't do handwriting recognition. It just digitizes your handwriting.
I see.
And then every now and then he would take a picture of one of your slides, and then he could annotate it. He could put notes in the margin, and it would go right into the document. So then at the end he had a long document with a lot of notes and slides here and there, which I thought was a really good record.
Wow.
That's cool.
Of the talk.
I'm astonished that he thought there was anything worth writing down.
Well, come on. It was a great talk.
Well, I had a good time.
No, I enjoyed it.
And I enjoyed Stefan. Stefan was a great guy. He was a great host. What a great time we had.
Yeah, we had to. Rich and I both gave talks. We did a TWiV. We had dinner with people, lunch, breakfast, talked virology the whole time. It was really good.
Good fun. Nice students.
Great
program.
Good program. Just too cold out there. When we left, it got warmer. And then
21 degrees when we got up on Friday.
Then the next day, they lost to Nebraska, right?
Oh, I didn't even follow that.
I heard. Alan, what do you have?
I have something that just became available a couple of months ago. The Royal Society has made part of their journal archive permanently free to access. Now, part of their archive, it's papers over 70 years old. The Royal Society has been around for quite a while. In fact, they published the very first peer-reviewed scientific journal of any kind in 1662. And you can read that online. That's the Philosophical Transactions, giving some account of the present undertakings, studies, and labours of the ingenious in many considerable parts of the world. So they started in 1662. They've been publishing since then. And you can go back to old papers, historically interesting papers from everything from the beginning to 70 years ago. Of course, be nice if they made stuff more recent accessible, but this is, I think, a big step in the right direction.
So
cool.
It's just from only up to 70 years ago, right?
Yes.
Okay.
Yeah. So if you want to read Charles Darwin's old papers or
sure.
Isaac Newton's,
I was going to look.
papers.
I was going to look up Darwin. I was just looking up Jenner, but he had to publish his original inquiry himself. Nobody would publish it.
Right.
Yeah, I think that's a great step. And then when they do the next 70 years, it'll be even better.
Yep.
Will they ever do that? Who knows? Cool. My pick is a post by David Tuller, who is a journalist, also a professor out at Berkeley School of Journalism. We talked to him on TWiV, I think it was 119. He's written a long post called Chronic Fatigue Syndrome and the CDC, A Long Tangled Tale. It's about 5,000 words. Longest post I've ever had, but he asked to post it on my blog, Virology blog, and it's there. And I know that a lot of people do visit the blog, but this is to point out David's wonderful piece on the history of what the CDC has or hasn't done about chronic fatigue syndrome. It's a really nice piece, and you should definitely check it out. It is long, but it's really good.
It is very thoroughly researched, and I can tell you that doing a piece of this depth is an enormous amount of work.
Huge, and I'm so honored that he wanted to put it on the blog. But
yeah,
it was a good idea. He's getting a lot of feedback, a lot of positive feedback, which is great. And it's kind of going to be the record of the whole story that people can say, oh, you should go read this, and then you'll know what went on. So,
great.
Yeah, it's not so much about the science as the people behind the science who, being people, they don't always behave the way you might want them to.
Right. And our listener pick of the week comes from Judy, our friend out in California, who's a high school teacher and who's written before. She sends an article in The Scientist, top 7 ranked articles in microbiology, according to the faculty of 1,000. And she writes, and I know about most of these from your shows. Thanks so much for the wonderful education. So there you go. Top 7. And pretty soon we'll have our 10 stories of 2011 coming up.
I've already started thinking about that.
Yeah, we'll put up an agenda so you can all start adding. So thanks for that, Judy. And that'll do it for TWiV 159. You can find us on iTunes, at the Zune Marketplace, at twiv.tv, and at microbeworld.org. And as I said, there is an app made by the ASM. It's also at microbeworld.org/app. Please send us your questions and comments. We love to get them. TWiV at twiv.tv. Alan Dove can be found at alandove.com. Thank you, Alan.
Always a pleasure.
Good to have you post-Thanksgiving.
Yes.
Appreciate it. And Rich Condit, usually at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. Great fun.
Well, you're going to be hanging out in Texas for a couple of days, right?
Going home on Sunday.
All right, enjoy. Hey, what's the weather like down there in Texas?
It's gorgeous. It's a drier climate, so it's been in the mid 70s during the day and down in the 50s at night. It's just gorgeous. It's beautiful.
Wonderful. Well, have a good time. And I appreciate you, both of you guys, doing this during the holidays.
Hey, I wouldn't miss it.
Good. I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.